My name's Seth Davis. I'm a postdoc with Sanford Eigenbrode and part of the Biotic team. What I'm going to be talking about today are long-term patterns of cereal aphid abundance in the Pacific Northwest. So the rationale for some of the analyses that I'm going to describe today are based off of the fact that there are really very few data sets that look at phylogeny and biogeography of aphids and herbivores of wheat systems over long time periods. And we wanted to use some of the information we had to try to predict how climate change might impact the population dynamics of these insects in the future. So this is an aphid, a rather grainy picture of an aphid. This is a picture of Rhopalosiphum paddi, the bird cherry oat aphid. It's an important pest of cereal grain crops and also a vector of some rather important viruses. This is the insect I'll be talking about today. We analyzed a number of species in our work, but for the purposes of time, we're just going to focus on this individual species for today. This is a suction trap. There is a landscape-level network of these things out there in the Pacific Northwest that just continually draw air in through the top during the wheat growing season and managed to sample ambient aphids just floating around in the atmosphere during certain times of the year. So you can actually think of aphid prevalence as something of an atmospheric event in some cases. In any case, these things are about 12 meters tall, and they were operated continuously for 17 years. So we have a rather large data set with which to investigate aphid population dynamics in the Pacific Northwest over this 17-year period. So using these data and some downscaled climatic projections, which we got in collaboration with John, we asked 4 questions. Our first question was asking whether or not aphid biogeography was structured by patterns of interannual variation in abundance. Our second question was asking whether or not there was evidence for a shift in the timing of peak aphid captures, that is, aphid phenology over the trapping period. Which environmental variables were most correlated with aphid trap captures over the trapping period. And finally, can we use variables like thermal sums and cumulative precipitation indices to model aphid abundances? So I'm just going to jump right into the analyses, so hold on because there's a lot of statistics that are about to get thrown at you. The first question, asking whether or not aphids are biogeographically structured by patterns of interannual variation, was investigated using a single linkage clustering algorithm. So this is essentially a multivariate technique for analyzing or grouping sites based on their variation in interannual aphid abundance. And you'll see that there's basically 3 pretty distinct clusters that broke out in this dendrogram, with a 4th site representing a 4th cluster that's just an outlier. In any case, this is a statistical basis for grouping these sites together, and to each of these colors corresponds a graph of the phenology, the average phenology over that 17-year period of the abundances of these aphids. So on the y-axis, and all these are Julian date. On the x-axis is a Julian date, and on the y-axis for all of these is a measure of aphid abundance. So you'll note that essentially phenological differences correspond to different clusters here. But we wondered whether or not these statistical clusters were in fact geographically supported. So if we look at the way those traps fall out on the landscape, again with these colors representing an individual trap and the statistical cluster with which it fell into, we essentially used a proximity analysis to analyze pairwise distances between these traps, asking the question, is the difference from, say, any red trap closer on average to other reds than to all other randomly selected points? And it turns out that for the cluster 1 and cluster 2, the red and the blue, there was in fact geographical support for those. That is to say that the average distance between all the red points was less than the average distance to all the other points. When we investigated our second question, looking for a shift in aphid phenologies over the trapping period, we found that there was pretty strong evidence for for peak aphid captures to come earlier over the trapping period. So in this first graph, here's year on the x and the Julian date of mean aphid captures on the Y pooled across all the sites. And you'll note that our linear regression essentially predicts peak aphid captures coming almost a month earlier by the end of the 17-year trapping period. And this was correlated here at the bottom with a slight overall increase in mean temperature projections for those locations. When we asked our third question, trying to get at which environmental variables were most strongly correlated with aphid abundances, we investigated mean temperature, thermal sums, cumulative precipitation, daily precipitation, wind speed, and wind direction. Each of the orange boxes is showing you a significant correlation. The correlation itself is shown in the box and for each cluster. So you'll note that generally mean temperature appeared to fairly uniformly affect aphid abundances, daily observations of aphid abundance. But other things like thermal sum, cumulative precipitation, and wind factors tended to be more cluster specific. That is to say, they were more region specific. Also, aphids tended to be strongly correlated with abundances of other aphid species. When we threw all this together in a big linear model looking at variables that represented climate space, thermal sum, and cumulative precipitation, we found that thermal sum itself was in fact the single strongest linear predictor of aphid abundance, making up for the vast majority of the sum of squares in this model. So we did a quick simulation experiment, randomly drawing from those factor distributions to generate model-level outputs, and we found that, in fact, simulated means of cumulative aphid abundances are strongly predicted by the accumulation of degree days. So essentially, the warmer it gets, the more aphids you get. So to summarize those questions,
single linkage clustering algorithms generated groupings based on interannual aphid abundance, and some of these groups tended to be geographically supported. Regression analyses suggested that peak aphid flights tended to occur earlier by the end of the trapping period, and this, interestingly, was also correlated with an increase in mean temperature in the region. Mean temperatures and interspecific aphid abundances were uniformly correlated with aphid abundance, but other factors tended to be cluster specific. And finally, linear mixed effects models indicated that thermal sum was the single best predictor of aphid abundance, and the simulated effect of thermal sum was strongly linear.
And I'm done. Thank you.